Okay, so yeah, so this was a question. It was a question about concentration. So it's about sodium fluoride, and it was talking about some toothpastes. Some toothpaste contains sodium fluoride, and the concentration of sodium fluoride can be expressed in parts per million. Now, what might have happened is I might have forgotten to do the very last step on the question, which it looks like on my answers, and I only got to the grams part and then didn't do the parts per million, which could be where we went wrong. So it was all the right process, and then I've just seen that I didn't do the parts per million, so that could be where it went wrong, but we'll talk it all through again. So basically, whenever we work anything out in parts per in concentrations, we often use it as parts per million just because we're using such small quantities. So in this instance, 1 part per million represents a concentration of 1 milligram in every 1 kilogram of toothpaste. So for example, if we had 1 milligram of sodium fluoride in a kilogram of toothpaste, we would then write it as 1 part per million. So basically, the question says that a 1 gram sample of toothpaste was found to contain 2.88 times 10 to the power of minus 5 moles of sodium fluoride. And you were asked to calculate the concentration of sodium fluoride in parts per million for this sample of toothpaste, and to give your answers into 3 significant figures. So let's have a go at working through this question. So the first thing you need to do is to work out the MR for sodium fluoride. So our MR, there we go. Our MR for sodium fluoride, which is our NaF, equals 42. So that's our first part that we need. Then we need to work out our mass. So once we've got our relative formula mass, then we need actually our actual mass. So we want to work out how much sodium fluoride is present in 1 gram. So we want the mass of sodium fluoride, NaF, in 1 gram. So to work this out, we would use this part. So we've got in 1 gram sample of toothpaste, we were found to contain 2.88 times 10 to the minus 5 moles of sodium fluoride. So basically we're going to convert these moles into grams. So we're converting moles into mass here. So we're going to use this value here of 2.88 times 10 to the minus 5 mole. And we're going to then multiply that by our MR, which is 42. That would then give us the answer of 1,210. So then we're going to do it in 1 kg. So in 1 kg, which is what we need, it would be 1.210 grams. So basically what I've done there is our answer here would have been 1.210 times 10 to the power of negative 3. And what we've done with that is because we want to change it from grams to kilograms, because the original question was asking us to talk about kilograms here. We need to convert this into kilograms. So now, so basically our answer, we would get our answer, we'd multiply our answer by 1,000 because we're talking about it in grams. Now converted to kilograms. In kilograms, we would say our mass of sodium fluoride in 1 kilogram would be 1.210 grams. So then finally, the last thing that we need to do is then convert it into parts per million. So from the information that we're given here, we're told that 1 part per million represents a concentration of 1 milligram in every 1 kilogram of toothpaste. Well, here we know we've got 1.210 grams in a kilogram. So we just need to convert this into milligrams. So we're going to convert that into milligrams. So again, we're going to times that by 1,000. And that would give us 1,210 milligrams. So to then convert that into parts per million, this is our last stage and nice and simple. 1 part per million represents a concentration of 1 milligram in every 1 kilogram of toothpaste. So this literally just converts straight into our parts per million. It goes into let's have a pen there. It becomes 1,210 parts per million, and we don't need to do anything else to that, and that will then be our final answer, 1,210 parts per million. I'm just going to take a screenshot of that and save that. Yeah, so that's sort of the step. I think what happened is I'd got to the grams part and then I'd done the foolish error of what sometimes happens in exams. I forgot to check that the question was asking me to give my answer in parts per million, not in grams. It can happen with concentration, so when they want it in parts per million. So I'd just forgotten to do that final step there, which is maybe where the confusion came across, because I forgot to do the final step. If we're talking about where the marks come in this, you would get, it's a 4-mark question. So you get 1 mark up here for working out the MR. We get a mark for working out the mass in 1 gram. We'd get mark for working out the mass. Oh, actually, where does the 3rd mark come from then? 4th mark. Mass from working out kilograms. Oh, yeah. Mass from working out MR, mass working out in kilogram, mark from working out the mass in kilograms, mark for working out the mass in 1 kg, and then finally a mark for converting it into parts per million. And that's where your 4 marks would come from. Again, we would use the process of error carried forward in this. So if for some reason you made an error in calculating your MR and you'd come up with something like 40, you would obviously lose that mark, but then as long as everything else that you'd put in place further on was calculated correctly, the error would carry forward, so you would lose 1 mark. But that would be fine. And again, at any point in these calculations, if you've made an error with the maths, you've calculated one thing wrong, as long as you then kept that error the same because you haven't realised it's an error, so you won't get 4 marks, but you can get up to 3 marks with error carried forward. So that's sort of how that works. So let me switch over now. So let me clear all that. So I hope that's nice and clear now. I'm going to actually look at infectious diseases because I've had an issue trying to get my Redox stuff to load, so we'll finish Redox tomorrow. So we'll do a bit of biology today and then we'll go back to Redox tomorrow because I was having issues finding my Redox stuff. So disease transmission, no, we've done this, so I don't know why I've got this one up. Give me a second, let me stop recording. Share screen. Okay, so where we've got to in biology, we've sort of covered immunology, we've covered some infectious diseases, so sort of pathogens as a whole, how immunology works, HIV and AIDS, so the different stages of HIV and AIDS, and then we're just going to have a quick look at malaria. So we use HIV and AIDS as an example of a virus, and then we use malaria as an example of a protist or a parasite. You can call them either. So let's go have a little look at how malaria is transmitted, the life cycle of malaria, how we can control malaria and global distribution of malaria. I'll have a look at TB and then have a quick look at antibiotics. And then we'll probably get a bit of time to move on to the next section. So I'll probably have a little look at lungs. I know we have done lungs, but there's a little bit about lungs that we haven't done. So malaria is caused by a protozoan parasite. At GCSE, we called it just a protist, but it's actually a parasite. Parasite meaning an organism that lives fully off another organism, and it causes detrimental harm to the organism that it's living off, and protozoan is the type of parasite it is. We get actually lots of different species of the parasite for malaria, but so we can have Plasmodium vivax, Plasmodium malaria, and Plasmodium falciparum. And the falciparum is the most common and the most dangerous type of malaria that we know of. It is transmitted by a vector, which is the Anopheles mosquito. Now, vector is really important. So most people say, oh, mosquitoes are what give you malaria. They do, but they transmit it. So the mosquitoes carry the parasite, but they are vector, they are what hold it, and then we become the host. It's very similar to the mechanisms of the Black Death with the plague, and how people thought it was a rat that was giving them. Yes, it was the rats, but it was actually a parasite that was living on the rats. So approximately 40% of the world's population, and generally those living in the poorest countries, are at risk of malaria. And without treatment, malaria can be very life-threatening. So what is parasitism? So like I've said, parasitism is a close and permanent association between two organisms. And basically one benefits from this relationship and the other one is harmed. And so there's no mutual reciprocation. It's literally one is taking and one is losing in this situation. So some parasites are pathogens that can cause diseases in humans. So we've got examples such as malaria. There's also African sleeping sickness, toxoplasmosis, and giardias. Parasites can be classified into endoparasites, and these live within the body of the host. So for example, a tapeworm, which can be found in the intestines, or ectoparasites, which you can probably think live on the surface of a host. So for example, a tick. Parasites can quite frequently affect household pets. So dogs and cats can have lots of issues with parasites. Things like fleas and ticks are massive issues. And also tapeworms. It's generally because things like cats and dogs, they don't have the same hygiene as us. They obviously don't wash their paws or their hands, and they eat lots of stuff that they find around. So it's really easy for them to ingest things like tapeworms and ticks. I mean, we can get ticks quite easily as well. It just depends where you go for a walk. So ticks live generally on livestock, so they can get passed from one animal to another, so to us potentially, or to dogs and cats and other livestock. So malaria has a range of symptoms, and these can include shivering, fever, sweating, joint pain, vomiting, and anemia. So if we think back to our original immune response, actually shivering and fever are quite normal. Our body temperatures rise to try and kill off any pathogens that enter our body. So you'll often find that actually lots of symptoms of different diseases are all very similar. And it's because of our generalized immune response. We get a fever, we shiver from the fever, we sweat because we've got a fever. Joint pain, vomiting, and anemia, they're the more where we start to suspect that it's malaria. The symptoms of malaria actually occur cyclically every 2 to 3 days, depending on which species of Plasmodium is involved. And that's because it's a parasite, and this is because it's closely linked to the life cycle of a parasite. So you don't actually necessarily feel ill constantly. It'll happen every 2 to 3 days, and you'll feel fine, and then you feel poorly again until your body really starts to get worn down. If untreated, severe malaria can lead to spleen and liver damage. And obviously our spleen is really important for our immune system, renal failure, comas, and death. So young children are especially vulnerable due to their immature immune system. So they have less of an immune system anyway. So they're more likely to not be able to fight off malaria. And actually, if they're trying to fight off malaria and get another illness at the same time, if malaria is affecting the spleen, that reduces their ability to then fight off another disease. So in a similar way, sort of like HIV and AIDS. So malaria parasite life cycle. So the life cycle of malarial parasite involves 3 important stages. So these are the sporozoite, the merozoite, and the gametocyte. And we're going to go through each of these 3 stages. So in stage 1, what happens is a female mosquito injects saliva and anticoagulants into a human host. The saliva contains the malarial parasites in the form of sporozoites. So the female mosquito, and it's only the female mosquito because this is to do with how mosquitoes reproduce, injects saliva and anticoagulants into a human host. It just happens that the parasites of malaria in the form of sporozoites are found in the saliva of the mosquitoes as well, so they get passed on to you as a human. So those sporozoites enter your liver cells. And when they enter your liver cells, they're able to multiply and they are able to develop into merozoites. At this point, these are then able to emerge from your liver. When they emerge from your liver, like when we spoke about viruses replicating, they burst out from your liver. They're damaging your liver cells. Once they've done that, they actually then go on and they invade your red blood cells. And when they invade your red blood cells, they actually feed and multiply inside your red blood cells as well. Then they emerge, bursting through 1,000 red blood cells. So again, we've damaged liver cells, and now we've gone on and we've damaged red blood cells. And this will cause fever in the host. If the host starts to notice that they're not feeling well, if your red blood cells are being damaged, you're not going to be getting enough oxygen around your body, you're going to start feeling very unwell. And it's going to lead to something like, it is going to lead to anemia because you've got a reduction in red blood cells. So, some merozoites will then go on and invade other red blood cells, causing more red blood cell damage. And some will actually then develop into gametocytes. And here we go, within the cells. So if the host is then bitten, so if you've already been bitten by a mosquito, you've already got the parasite in you, if you are then bitten by a vector, these cells can then be taken up in the blood meal. So another mosquito comes along, it bites you, and the cells can then be taken up that contain the parasite in the blood meal, which is then how we can pass it. Malaria can be passed from person to person. In the mosquito, the gameocytes develop into gametes. So the replication, the reproduction of the parasites happens inside the mosquito. So the male and female gametes of the parasite inside the mosquito fuse to form a sporozoite again, which migrates to the salivary glands of the mosquito to then start that whole process and cycle again. The sporozoites are now ready to be transmitted to another host in the mosquito's next blood meal, and the whole cycle can then begin again. So in the 1960s, the incidence of malaria was greatly reduced by the use of insecticides and drugs. However, the evolution of resistance in mosquitoes and Plasmodium species has caused a resurgence of the disease. It happens in a very similar way to the fact that we have different variants of viruses, because viruses mutate and become resistant, and the same way the bacteria does, that we get multiple, like MRSA, so your multiple resistant strains of your different bacteria. So this has caused a bit of an issue. So currently we use lots of different methods of control and prevention. And it's actually, there's a big push on prevention. There's lots of illnesses, but especially for malaria, we can cure it, but actually, if we can prevent it, that's a lot better. We will not having to try and cure as many people, and actually the less people we're having to treat means that we're giving the parasite less chance for it to evolve and become resistant to the different medications we're treating the people with. So the Anopheles mosquito can actually be controlled using insecticides, because that will obviously kill the mosquito, which means that the parasite inside it will die with it because we're killing the vector. So it's all actually about trying to prevent the vector reaching us. Environmental management and biological control. So a genetically modified malaria resistant strain of mosquito has also been developed. So this might help control malaria under the idea that if we release them into the wild and they bred together, eventually what would happen is these malaria resistant mosquitoes would hopefully become the dominant individuals variant in the communities that are there, and so they will be able, so we'd have less transmission of malaria to individuals, and then hopefully the mosquitoes that are able to carry malaria would slowly start to die out. So malaria is most common in the world's equatorial regions, and that's because of the prevalence of malaria is actually linked to climate, and that's all to do with mosquitoes. So malarial parasites in the mosquito actually develop a lot quicker at higher temperatures. It's why somewhere like England or Canada or Iceland, for example, it'd be very rare, if completely unlikely, to be able to get malaria because we wouldn't have high enough temperatures for the parasite to develop quickly enough. And that's because the lifespan of a mosquito is linked to the temperature and the humidity. And mosquitoes need water to breathe. So when we were talking about prevention before, one of the biological controls and environmental controls is like mosquito nets placed over beds of water. And that's actually to stop and try and prevent mosquitoes from being able to breed in the water, which then should decrease the mosquito populations. So therefore, because of these situations, malaria is most common in warm, wet regions. So climate change could actually affect the number of people at risk for malaria. If we think about global temperatures rising, that band of where in the world it's warm enough for mosquitoes to breed and live would actually spread up across the world. So I'm going to have a quick look at that now. So climate change and malaria, we're going to look at the scenario for 2050. So this is our current distribution. So if we think we've got the equator just around here, this is all our equatorial regions. If climate change continues and global temperatures rise, what we think could happen is that we then get these extra extended distribution, these extra countries. So if we look here, we've actually got quite a lot of the southern states of North America being affected, and all these extra countries and places being affected by potential malaria that wouldn't be before, places like Australia. And again, it's only certain specific regions of them, and I'll explain why now. It's just because of winds and other climate issues. So lots of coastal regions, as you can see, sort of here, if we go up, so we have got coastal regions here, but some of the coastal regions in North America wouldn't be included in that because necessarily apart from the sea, there might not be enough other lakes and pools in those areas, so there wouldn't be necessarily somewhere for mosquitoes to breed because they don't, whilst they need to breed in water, it needs to be fresh water, not the sea. So the life cycle of malaria, we have done this, so let's have a quick look at HIV. Did that. That's up twice for some reason. Right, where's what I want? TB. Okay, so we're going to have a quick look at what TB is, what the infection is, and the symptoms and causes of TB. So TB, tuberculosis, is caused by an airborne bacteria, Mycobacterium tuberculosis, and it is a widespread disease that affects the respiratory system. So this is an image of the TB bacteria, and 1/3 of the world's population is currently infected with the bacteria. It's not something we talk about huge amounts in the UK because up until, I'm trying to think now, most people used to be vaccinated against it. When they became 13 at school, you would normally get a vaccine against it. However, I'm trying to think how old I am, how many, about 12 years ago, and they stopped doing that because they said there was enough population immunised against TB. So using the idea of herd immunity, we wouldn't need to immunise any more of the population. However, there's lots of arguments about that because obviously as the amount of older generation dies and we get a growing younger population, we'll have more people who aren't immunised against TB. And obviously, it says here 1/3 of the world's population is currently infected with the bacteria. Whilst it's very rare in the UK, with more immigration and emigration, and more travel of just for holidays, more people are going to be likely to come into potentially into contact with TB. Before, if someone went on holiday to a country that might have TB, the chances of anyone going who wasn't vaccinated would be low. Now those chances are increased, so we're now having to vaccinate people if they go on holiday to certain countries, just because TB is more prevalent there. If you work with certain communities of people in the UK, so homeless people, and people in prisons, generally because often they haven't been either in school system, or they just haven't been and gotten their vaccinations when it was time for them, and the sort of environments that they're living in close quarters with other people, who are likely to have TB, means that TB is quite a prevalent disease in those communities in this country. So we do still vaccinate against it, but it's more specific people that we vaccinate against. So specific jobs, anyone working, for example, in the NHS would be vaccinated against it. So we do still vaccinate, but it just depends. And actually, when we vaccinate against it, we check we check that we actually don't have an immunity to it first, and then we give the injection. So TB can be treated by antibiotics, but again, we had lots of antibiotic resistant strains of TB have emerged, so that's when this real big push on actually using immunization came in, because actually it's that idea of prevention is better than cure. Cure was causing these resistant strains to emerge, so if we can prevent people from becoming ill with TB, that's a lot better than trying to cure people. So what is a tuberculosis infection? So when a person gets infected with TB, an immune response is produced. In healthy people, white blood cells called macrophages, that we've talked about before, they come along and they engulf the bacteria by phagocytosis, controlling the infection. So like we said before, they come along, engulf it, they release all these different enzymes, they digest it, and the bacteria is killed, it was destroyed, and the body gets back to normal. So a tissue mass called a tubercle forms around the infected site, and after 3 to 8 weeks, the infected region heals. However, the bacteria can survive inside macrophages for years until the immune system becomes weak, and this is a massive issue with TB. So macrophages engulf the bacteria by phagocytosis and control the infection, but we're not able to actually rid it fully of the TB bacterium, and they can actually survive inside the macrophages for quite a long time until the immune system becomes weak. It sort of stays hidden for a while. And the second that the immune system becomes weak, then the whole infection sort of rises again, and it has a second wind, again, very similar to the process of HIV and AIDS. So symptoms of TB: if a patient's immune system is unable to control the infection, active TB occurs. So this point is inactive TB. You've got the infection, but you're not actually in active TB at the moment. Healthy people, this might actually happen, and you might not ever get a resurgence of the TB. But if your immune system becomes weakened for some reason, you could pick up another illness, you could become pregnant, have another disease, then active tuberculosis may occur. Bacteria then will multiply rapidly and destroy the lung tissue, which can be fatal. So a patient with active TB may experience symptoms such as coughing, shortness of breath, loss of appetite, weight loss, night sweats, fevers, and extreme fatigue. Fever and night sweats occur due to neutrophils and macrophages releasing fever-causing substances as part of that inflammatory response. So again, it's that generalized inflammatory response of when you're ill. These chemicals affect the hypothalamus and increases our core body temperature. The issue is, if our core body temperature remains too high for a long period of time, that has other damaging effects on our body. If we think back to enzymes have an optimum temperature, but we work out that temperature becomes too high, this enzymes denature. If we have a fever and sweats for too long, high temperature for too long, enzymes in our body will start to denature. Our natural bodily processes start to not work properly because we don't have enough enzymes for those processes to take place. So cause of TB, is there a correlation between TB and poverty? So this is our TB incidence up here in countries, and the basically the amount of money in thousands of dollars spread out per person, so GDP. So in countries where we're talking between $0 to $5,000 per person each year, the incident rate of TB is highest, and we see that then there is a decline down to less and less the more prosperous and affluent a country is. So now we're going to have a quick look at antibiotics. So we've talked a lot about diseases that can be treated with antibiotics, but what are antibiotics? So antibiotics and how they work, how antibiotics can be tested, and look at antibiotic resistance, because we've mentioned it a lot but not really spoken about it. So antibiotics are a group of drugs that we use to treat bacterial infections. They cannot treat viral infections, only bacterial infections. And so they're effective against prokaryotic cells. They generally leave eukaryotic cells unharmed, and that's really important. So bacteria is a prokaryote. Bacteria is eukaryotic cells. So antibiotics are great because they generally destroy and damage prokaryotic cells, and they leave all our eukaryotic cells in our body completely unharmed. Bearing in mind our whole body's made up of eukaryotic cells, antibiotics would be really not great if they damaged all of our eukaryotic cells. Now, we do use the term generally because obviously there can be instances and occasions when they can get slightly harmed during the use of them. And we have two different types of antibiotics. So we have bactericidal, and these go in and kill the bacterial cells. And then we have bacteriostatic, and these slow the growth or reproduction of the bacteria. So then your own immune system is then able to take over and remove the bacteria itself. Some antibiotics prevent the formation of bacterial cell walls, and this results in osmotic lysis. So testing antibiotics. So actually, people create antibiotics, but we need to know actually how effective they are, how do they work. So what we're going to look at is how we test the effectiveness of antibiotics against a specific type of bacteria. So what we do is a sterile swab is taken and is used to inoculate an agar plate with one type of bacteria. You may have remembered this practical, you might have done one very similar at GCSE, and you take a swab of a bacterium, for example. So this would be one specific type of bacteria. We take a sterile swab of that bacteria, and then we inoculate the agar plate with that type of bacteria. So that agar plate has a growth medium on it, which allows the bacteria to grow and live and form a bacterial colony on it. What we then will go and do is we get paper discs. And we get some paper discs, and they contain different types of antibiotics. And we place these on the agar dish. So we're growing the bacteria, we get these paper discs containing different types of antibiotics, and we place them onto the agar tray. We place them out generally very symmetrically. We'd normally only do no more than 4, with equal distances between them. We then seal our plate and incubate for 24 hours at 37 degrees. Now we say we seal it, we do seal it, we don't necessarily seal it fully. We do often leave a little bit of an air gap, and that's to stop anaerobic bacteria growing. And we incubate it for 24 hours at 37 degrees. If this was happening in the school environment, it wouldn't be 37 degrees because, again, the sort of bacteria that can grow at 37 degrees can be quite harmful. And schools just don't have the classification, or colleges have the classification to deal with that sort of biohazard. But in general, in a lab, we would seal it, incubate it in an incubator, which is like a fridge oven. So it's obviously not as hot as an oven, but it's not as cold as a fridge. It's 37 degrees, we can set them, but they look a bit like a fridgy oven. And we leave them in there for 24 hours. So we seal it, and there you go. There's a small one which you can get them that are as big as a fridge freezer. And then we look at them after 24 hours. So after 24 hours, we look at the plate, and we look at actually the diameter of the clear zones that are measured around the circle. So basically we call this the zone of inhibition. So these areas where we've got no bacterial growth around each of the paper discs are called our zone of inhibition. So for example, C has absolutely no zone of inhibition. So we would record that one as 0. I think it's going to just do that for all of them. So I'm just going to skip to them. It's got all of them. There we go. And just from looking at them straight away, we can start to suggest, well, which antibiotic is the best, which one's the strongest? And from this, we go, well, D. D's got the largest zone of inhibition. It's got the largest area where bacteria wasn't able to grow. The worst one was C. It didn't stop the bacteria growing at all. And then it would be, D would be the best, followed by B, A, and then C being the worst. Just like that. And so then what we could do is we could repeat using different species of bacteria. So actually we could say, okay, antibiotic D is great against bacteria 1, but can't you see it great on number 2? But D, I think, is the best on bacteria 2. Maybe, yeah, D. And then A and B, C did nothing. Oh no, C did a little bit. We could possibly suggest that neither of them are really great contenders for bacteria 2. Bacteria 3, none of them work. So we say we'll have to look for a different type of antibiotic for them. And I don't even know what's happening on this now. And then bacteria 4, A would be the best. So which antibiotic is most effective against this type of bacteria? Well, looking at that, we would say straight away D. And then this experiment could be repeated using different species of bacteria. Which species of bacteria do you think has developed resistance to the most antibiotics? Well, that from this image would be bacteria 3, because there is absolutely no zone of inhibitions around any of those paper discs. So we've mentioned antibiotic resistance, but what actually is it? So in the presence of an antibiotic, bacteria of resistance to that antibiotic, and that is due to a genetic mutation in the evolution, have an advantage. They are able to survive and reproduce, giving rise to a population of antibiotic-resistant bacteria. And in some cases, this resistance is evolving faster than the development of new antibiotics. Tuberculosis has evolved resistance to streptomycin and other antibiotics, and so now we have something called a multi-drug resistant TB. It's still relatively rare, but there is growing concern about its spread. So again, it's why prevention, inoculating people against TB is often preferred to letting people get TB and then trying to treat them. It's why there's always lots in the press, especially when you go to doctors and you're given antibiotics and you're told you must take your full course of antibiotics. And it's really important because basically if you don't take the full course, what often happens is you can start to feel better. And sometimes antibiotics have some bit rubbish side effects, so you can feel a bit unwell as well from them. And you can start to feel better and be like, oh, my antibiotics make me feel worse, I'm going to stop taking them. But actually what might have happened is that not all of the bacteria has been killed. So what can then happen is you can have this resurgence of the infection. But during that time, bacteria can mutate, and bacteria can mutate and become resistant. And then you have a second infection that's where more of the resistant bacteria remain, able to fight. And then if you're given the same antibiotics, it's not able to fight it off anymore. So you have to be given stronger antibiotics. And then the same thing again, then you need stronger and stronger. And so then looking at antibiotic resistance, so bacteria Staphylococcus aureus has become resistant to most antibiotics, that's MRSA. So that's including methicillin. So we get methicillin-resistant Staphylococcus aureus, MRSA, and it's very common in hospitals and can cause a life-threatening infection. So due to the emergence of antibiotic resistance, we have several methods of prevention and control being adopted. So antibiotics should only be prescribed when needed. So doctors now are very, very hesitant to prescribe antibiotics unless it's really clear they're needed. In the past, when they were quite new, doctors would just give them out because we didn't realise about this resistance issue. And so it was like, oh, you're ill, you've got sore throat, yep, we'll give it you. Whereas actually now doctors are a lot more hesitant to give you them, and they'll be like, no, actually wait, see how you're feeling. If you're still not feeling better, we'll maybe do a swab and then we'll give you some antibiotics. Because we can't tell just from symptoms are different often if you've got a virus or if you've got a bacteria. Bacterial infection, if there's a virus, antibiotics aren't going to do anything, but they can increase to the antibiotic resistance. Again, if it's just a mild infection, do we really need to be giving antibiotics out? If you're a young, healthy adult, you can probably fight that infection off yourself rather than adding to the antibiotic resistance issue. Often young, healthy adults are the ones who don't finish the antibiotic course because they go, oh yeah, I'm feeling fine. That's where these issues arise. And so patients should finish a complete course of antibiotics and introduction of infection controls in hospitals. So the pathogen, Staphylococcus aureus, is resistant to methicillin and multi-drugs. So in US and Japan, and it is where in the US it compromises 32% of drug-resistant infections, and in Japan it compromises 60% of those infections. If you look at TB, it's we've got resistance to it of any drug at all. Is completely resistant to any drug, and that's found in the US, and that compromises 13% of their drug-resistant infections. And in New York City, that's an even higher level at 16%. And in Eastern Europe, they've got a multi-drug TB resistant strain. It's not any drug, it's just multi-drug. It's getting closer to any drug. Then we have Plasmodium falciparum, which obviously is another type of bacteria, and the drug that is resistant to is chloroquine, and this is in countries such as Kenya, Ghana, Zimbabwe, and Thailand. Again, the drug-resistant infections, they compromise things such as 65, 45, 59, and 45%, respectively, of the prior list. And then we have Streptococcus pneumoniae, which is our general pneumonia. And we have lots of penicillin-resistant strains of this. So USA, Asia, and Chile, it's between 10 and 35% of all their drug-resistant infections. Spain, 20%, and Hungary, 58%. So, let me just, oops, get this up quickly. I don't want plant transport at the minute. Organisms, gas exchange. So we've talked about gas exchange. I'm going to have a quick look at ventilation, because we didn't really do that before. So it leads on quite nice because we were just talking about TB. So we've looked at the lung structure before in the past. If you can't remember it too well, have another look your own time in a bit, maybe go back on one of the other videos that I've done, have a look at lung structure. But we're just going to have a look at ventilation now. So we've done gas exchange, done the lung structure, but actually ventilation. So we're looking at the mechanism of ventilation, the role of the pleural cavity, composition of inhaled and exhaled air, and measuring lung capacity. So animals need to maintain a concentration gradient across their exchange surfaces so that that oxygen will diffuse into the blood and carbon dioxide will diffuse out. So we've spoken about it before, we have to have that concentration gradient. So we have that continual diffusion of oxygen and carbon dioxide in either direction. And we always say, oh yeah, it has such and such and such and such to maintain a steep concentration gradient. So fish manage this because they have a continuous stream of oxygenated water moving over their gills, and we'll look at fish in a bit more detail in another session. But in animals such as mammals and birds, we actually have to have a concentration gradient maintained in our alveoli by a mechanism called ventilation. So what is ventilation? So ventilation is basically the process of inhalation and exhalation. And this is where younger children get confused with respiration being breathing. Ventilation is inhalation, exhalation, breathing, and respiration is obviously to do with that production of ATP. So muscles and bones of the thoracic chest cavity work together to increase and decrease the size of the lungs during breathing. Nothing too complicated here, probably nothing that you haven't heard before. So we've got the diaphragm, this large sheet of muscle that sits underneath our lungs. And what happens to the diaphragm is during inhalation, the diaphragm contracts. And when it contracts, it flattens in shape and it allows our chest cavity size to increase. So during passive exhalation, it relaxes and it allows our lungs to shrink. So during passive exhalation, it relaxes, and during inhalation, it contracts to increase. We also have our rib cage, and our rib cage does a lot more than just protect our internal organs. So our rib cage lifts up and out during inhalation, and this allows us to increase the volume of our chest cavity because the rib cage is restrictive of how much airflow we could get in. So if we lift our ribs up and out during inhalation, we increase the volume of our chest cavity. The first and 12th ribs are fixed, but the others are able to pivot between them according to the action of the intercostal muscles. So you can see here, the first rib is fixed and the 12 ribs are fixed, but the others have intercostal muscles at the side of them, so they are able to pivot slightly. So then we have our external intercostal muscles. So the external intercostal muscles contract and they pull the ribs upwards during inhalation. And the first band is anchored to the first rib here, and it is fixed, and each subsequent band lifts the next rib up from above it. And then we have our internal intercostal muscles. And our internal intercostal muscles contract to pull our ribs downwards during forced exhalation. And the lowest band is anchored, as you can probably guess now, the opposite to the last 12th rib, which is fixed, and each pulls the next rib down from below. The only thing we haven't mentioned then is the pleural cavity. So each of our lungs is actually enclosed in a double membrane, very similar to when we talk about the heart and it's in the pericardium. The lungs are in a double membrane known as the pleural membrane. And the space between the two membranes is called the pleural cavity. And it's filled with a really, really small amount of pleural fluid. So here we've got our lung and we've got our pleural membranes on either side. So our lungs here, and I don't like that bit, yeah. Our lungs here, it's our pleural membrane, pleural fluid and pleural membrane again. And what this fluid does is it lubricates the lungs. So we've spoken about the advantages of the lungs for gas exchange and the fact that they're lubricated. So it adheres the outer walls of our lungs to the thoracic chest cavity by water cohesion. So we've spoken about water cohesion before because of the polarity of water. So the lungs are able to expand with the chest while breathing. So the lungs, because of this electronegativity of water, the polarity of it, we can have that movement happening. Oops, was that it? Was it? So in one breathing cycle, the air in our lungs loses only some of its oxygen content. And so this is why mouth-to-mouth resuscitation can be effective. So basically, we're going to look at the composition of inhaled and exhaled air. So this is nitrogen. Nitrogen makes up 78% of our inhaled and our exhaled air. Of our inhaled air, oxygen comprises 21%, and it comprises 15% of our exhaled air. Our inhaled air, we do actually breathe in a small amount of carbon dioxide. We breathe in 0.04, well, yeah, bring it back in. We exhale 4%. Water, we have less than 1% inhaled, but we'll exhale around 3%, and then we have other gases as well. So this is really important. So actually, whilst we inhale 21% and of our inhaled gases, it's 21%. We actually look, it's only actually a small amount that will stay in there. Lots of it we breathe in, but because of the way diffusion works, actually when we breathe out, we're actually letting out quite a large amount of oxygen as well. So we can measure someone's lung capacity, and we use something called a spirometer for this. And this is a device that measures the volume of air passing through it during breathing. And its results can be displayed on a kymograph trace. So we're just going to look at the measurements of this. It looks quite similar to an ECG, but it's not, but it sort of just looks at our lung volume. So we can measure our total lung capacity, so that's how much air our lungs can contain the most. So total lung capacity is total volume of air the lungs can hold after a maximal inhalation. So that's the largest inhalation we can make. So in an average size 70 kilogram adult male, this is around 6 litres. So then we also have our tidal volume. So we've got our lung volume, so our largest lung capacity. So we also have a tidal volume. So during relaxed breathing, so that's generally your natural breathing, non-forced inhalation, exhalation, only a small part of the lung's total capacity is actually replaced with each breath. The volume inhaled and exhaled is known as a tidal volume. And in an average sized adult male, this is about 0.5 litres. So if we think our total lung capacity in average male would be 6 litres, this large area here, this area here, our tidal volume is actually on average only about 0.5 litres. And then we can look at our inspiratory capacity. So the maximum possible volume of inhaled air after a relaxed exhalation is known as the inspiratory capacity. The accuracy of this requires the patient to breathe out as much as possible. And in an average sized adult male, this is about 4 litres. That's a bit of a strange one because we think, well, that's less than our total lung capacity, but that's because we've breathed out as much as possible and we're taking everything in. As we said before, we don't ever really often breathe everything out. We often keep quite a lot of our gas inside our chest, and that's when we have our tidal volume. As you can guess, our expiratory capacity is the opposite. So it's maximum possible volume of exhaled air after relaxed inhalation. And this is known as the expiratory capacity. So the accuracy of this requires the patient to breathe out as much as possible. So they're often what we use for a peak flow monitor for someone who's got asthma. There's someone to do a normal breath in, deep breath in, and then breathe out as much as possible. And then we have vital capacity. So this is a maximum volume of air that can be exhaled after a maximal inhalation, and that's our vital capacity. So if we took the deepest breath in and inhaled and exhaled our biggest breath out, what would that be? So in an average sized male, this is about 4.6 litres. This value is really useful when diagnosing respiratory problems. So if we're trying to work out if someone has something like COPD, for example. We also have a residual volume, and this is the amount of air left in the lungs after maximal exhalation. It is the capacity of the airways and the alveoli when deflated, and it cannot be measured with a spirometer. In an average sized male, this is about 1 to 2 litres. So what I'm going to do is I'm going to leave that there for today, otherwise